So before you go to SGD with these chapters, I want you to be thorough with the enzymes, enzyme defects, and the clinical features of these particular diseases. This is very, very important as far as examination is concerned, and even in your day-to-day clinical practice. So today we will start another important pathway. That pathway is never asked nowadays, but the products and the associated things to this pathway are very important as far as clinical aspects are concerned. So pentose phosphate pathway is also known as a secondary pathway. See this, the large fountain, or what do you call it, a cascade, waterfalls, shows a major stream of the water which consumes most of the amount of the glucose present in the cell. Pentose phosphate occurs side by side. Water is a beautiful thing. It is the best expectorant so far discovered, best expectorant, and the best fluid or liquid to make you awake, particularly cold water. Okay, just keep standing for some time, it will go on. It happens. So the major pathway which utilizes glucose is glycolysis. The side by side, the accessory pathways are there. One of them is pentose phosphate pathway, which also utilizes glucose. But this pathway, it originates from the glycolytic pathway, and it also goes into it also converge with glycolytic pathway. When it starts from glycolytic pathway, ends at glycolytic pathway. So this is a connection, right? Alternate connection. What do you call this alternate connection? Shunt. That's why this pathway is also called shunt pathway. So now this pathway produces pentose phosphates, so it is called pentose phosphate pathway. This pathway also produces hexose phosphate. So hexose monophosphate shunt pathway, HMP. Hexose monophosphate shunt pathway, HMP. Okay? Both the names are. The first intermediate of this pathway is gluconate, phosphogluconic acid. So then another name is phosphogluconate pathway. Okay? So these are the three names, alternate names for pentose phosphate pathway or HMP shunt pathway. It occurs in, all right, it occurs in all living cells. Unlike TC cycle. Does TC cycle occur in all living cells? Does TC cycle occur in all living cells? No. Which cell? Mature RBC, which has no mitochondria. So here we don't need mitochondria. We need cytosol only. Correct? Now the beginning of the pathway has the one intermediate called glucose 6-phosphate. That is the first product of glycolytic pathway, glucose to glucose 6-phosphate. This pathway begins with glucose 6-phosphate and ends at different regions of the glycolytic pathway. Sit down. Here we have shown the different fates of glucose 6-phosphate. Just go through it, I will not teach now. Straight away we will go to the pathway. Now if I start the pathway like this, there will be a problem after this step. That is, the number of carbon atoms count will be jeopardized. Why? Because this pathway requires 3 glucose 6-phosphate to start with. Each pathway will follow this pathway, each glucose 6-phosphate. But we have to start with 3 glucose 6-phosphate. So side by side, I will draw here, and my suggestion will be, if you do not understand and memorize the pathway here in this classroom, I may have to take another class for the same. No one so far followed my instruction in all these years. Correct? I hope you will also follow the same. But if you can break my hope, then you will be benefited. So my suggestion will be to be very concentrated, focused on how I am drawing and approaching with this pathway. To remember this pathway is very difficult, I tell you. But if you remember this pathway, you know how to remember, then it will be easy. So as I told, we will start three molecules of glucose 6-phosphate. What is the first reaction? The first reaction is catalyzed by G6PDH. Write down. This is very, very important. G6PDH. This particular enzyme is clinically important. G6PDH. What does it do? Oh, it is na. It na. Sorry, that was the wrong slide. This slide we must insert. Okay. So, see, the first one, first step, catalyzed by, utter with me. If you are not with me, you cannot remember ever, I tell you. If you want to remember it now, do it now. If you go back to your room, find out the same thing from your book or the copy, it will be very difficult for you to understand, I tell you. The way I will tell you, that is the easiest way to remember this pathway, so far, to my knowledge. I cannot be superlative in every case. Now, find it. So the first step is glucose 6-PDH, and this step produces one plus H plus. If I write NADPH2, then it is, or if I write NADPH plus H plus, which one is correct? If both are correct, then where are the uses? Any idea? You go to plant, you find NADPH2, plant system. You find it in animal system, it is NADPH plus H plus, correct? Like that. So, this particular step is similar for all the three glucose molecules. Correct? What is the product? See, it is taking up 2 electrons. That means this is what? Glucose 6-phosphate, this is aldehyde. So, if you take up electron, then what will happen? Aldehyde, see what is the product? 6-phospho, gluconolactone. 6-phosphogluconolactone. What is lactone? It is a ring structure. It takes up 2 electrons and there is a ring formed between C1 and C4. Gluconolactone. So, lactone ring. Correct? One C-double-bond-O is there in the ring and then a ring is formed. This lactone ring is broken down by lactonase enzyme. What is the name of the enzyme? Delta lactonase. Delta lactonase. Kya dekho? See, removal of, see, one molecule of water will go inside. Water will give what? OH. So the C-double-bond-O will become now HOH. So again the ring will be broken and a straight chain sugar will be formed. That will form 6-phosphogluconate. Now this was aldehyde, this is acid. So what happened? Oxidation. Right? For all 3 reactions same. Here we require 1 molecule of water. This is nothing but one hydrolase, but intramolecular. It is not breaking in two molecules. Within the same molecule, it is just breaking the lactone ring. Lactone ring. Next, the third step is similar to the first step. Third step is similar to first step. Why? Again, the enzyme is dehydrogenase, followed by the name of the substrate. It was glucose 6-phosphate dehydrogenase. Name of the substrate, dehydrogenase. This is 6-phosphogluconate dehydrogenase. Name of the substrate followed by dehydrogenase. So the enzyme is six-phosphogluconate dehydrogenase. As I told, this is the same thing like the first one. So NADP will form NADPH plus H plus. But is something else formed in comparison to step 1. What is that? There is a removal of 1 molecule of CO2. So now the 6-membered ring has become 5-membered ring. And the product is ribulose 5-phosphate. Write very clearly, or you can write like here also. Ribulose 5-phosphate, you write like this, okay? Here you have to write 3 times. From here you have to write 3 times. Previously I just put it in a single block. But from here onwards, we have to write discretely, separate names. Same thing, ribulose 5-phosphate, ribulose 5-phosphate, ribulose 5-phosphate. So we started with how many carbon? correct? After the end of first 3 steps, we are with the first 3 steps of this pathway is called oxidative phase. Oxidative phase এর পরে যে 3টে স্টেপ সেটা হবে rearrangement step phase. Reduction of number of carbon will be up to this step. Beyond this step, there will be only just rearrangement. So total number of carbon will be same. We'll find out. Correct? So these first 3 steps in many books, they have reduced it to 2. How? They have merged this one and this one. Sorry. In many books, they have merged these 2 steps. First and second step, they have merged to make it a single one. And they omitted the delta lactose step. Many books you will find. But remember, first and third, if delta lactose is there, otherwise first and second, these 2 steps only are the only 2 steps in our whole body which are responsible for production of NADPH. The NADPH production is very, very crucial for our livelihood, for us to survive. Without NADPH, we will die due to hemolysis. RBC will be lysed, cholesterol will not be synthesized, fatty acid will not be synthesized, nothing will happen. All the cells will undergo lysis without NADPH. It is so, so important, and there are only 2 steps. That is why when I mentioned this picture, it is actually a misnomer. What image that does actually it actually produces in your mind regarding HMP shunt pathway? It is a secondary, subsidiary pathway, correct? But no, when it comes to survival of the cell, it is primary. The cell can survive without energy for some time. Small amount of energy, the cell will starve, but it will not die. Then it will fast for some time. This pentose phosphate pathway is so important that 6 out of 10 glucose molecules goes to this pathway. If there are 10 glucose molecules inside the cell in the form of glucose 6-phosphate, 6 will go in erythrocyte, 6, if not more, will go to this pentose phosphate pathway, and only 4 will follow the glycolytic pathway. Correct? That is why it is so important. In other tissues, it is opposite. The 7 will go for glycolytic pathway, 3 will go for pentose pathway. Again, in different tissues, it varies. If the tissues which are concerned tissue is important for production of steroid hormones, cholesterol, then they will need more and more HMP shunt pathway to perform. Am I clear? So this is so important. So up to ribulose 5-phosphate we have come. Now you continue in your copy. I have to write it again from the top. So, what was that? Only I can reach that height. है ना? Only I can reach that height. What is, अच्छा. Now, let's see, there are three converging reactions, है ना? Converging reactions. Now here we have to remember this mnemonic. For last three reaction, we remember this mnemonic: keto, aldo, keto. Keto, aldo, keto. But see, before this converging reaction, there is another reaction which is converting them for same pentose phosphate. So remember these two. For next reaction, we have to remember AP, iso, AP. For last three reaction, keto, aldo, keto. What is that? Epimerase, isomerase, epimerase. Epimerase, isomerase, epimerase. Hello? This is one reaction. Next three reactions are same. Transketolase, transaldolase, transketolase. Transketolase, transaldolase, transketolase. Okay? AP, iso, AP, keto, aldo, keto. AP, iso, AP, keto, aldo, keto. So now AP, iso, AP. So epimerase, isomerase, epimerase. Ribulose, epimerase means what? They will differ in only one carbon place. So it will become remember not xylose, xylulose, ribulose, right? The isomerase will convert it into ribulose is ketopentose, ribose is aldopentose, correct? Correct? See, now this thing you cannot make out with this. You know? So, 2 out of 3 will be converted to xylulose 5-phosphate. The middle one will be converted to corresponding aldose sugar, ribose 5-phosphate. Then what are the name of the enzymes I said? AP, iso, AP. Then Done? What next? Remember, point number 2. Transketolase, wherever you have transketolase, you have a fixed coenzyme. Like transaminase has a fixed coenzyme. What's that? PLP, P-deoxyl phosphate. Carboxylation has a fixed coenzyme. What is that? Biotin. Similarly, transketolase has a fixed coenzyme called TPP, thiamine pyrophosphate. Now, transketolase. Trans means transfer, keto group. So there are 2 5-carbon containing compound. Which one is keto group here? Which one has keto group? Ulose or keto sugar. Which one? Xylulose. So which one will transfer keto group from where to where? From xylulose to ribose. So 5 minus 2 how much? 3. 5 plus 2 how much? 7. What is your group is transferring? Keto. So the 7th group will have the keto group. 7 carbon containing compound will have the keto group. 3 carbon containing compound will not have keto group because the keto group has been transferred. Right? So the keto group from xylulose will be transferred onto ribose to form pseudoheptulose 7-phosphate, and this will be glyceraldehyde 3-phosphate. Clear? 5 plus 2, 7, 5 minus 2, 3. Again, now transaldolase. Transaldolase does not require any coenzyme. It will transfer keto. Keto group always remain at C2. So transketolase will transfer 2 carbon. Transaldolase rather will transfer 3 carbon. So 7 minus 3. Can you deduct 3 from 3? No, it will be 0. So you have to deduct 3 from the 7. So the 7 will become erythrose 4-phosphate. 3 will go to 3. So 3 plus 3 what will be? 6. What is transferring? Keto group, the first part, first 3 group. Now glyceraldehyde will become a keto sugar. 3 plus 3, 6, fructose 6-phosphate, and these will become erythrose 4-phosphate. Erythrose 4-phosphate. Now it is started becoming complicated, na? Now you have lost your confidence about your concentration, I think. Huh? Are you with me or you are left? No, you are not with me. You can't be. At your age, it is not possible. But I would ask you to bring your mind focused on this. Otherwise, if you just lose your concentration here, you lose everything. The last step is transketolase will transfer what? C2, 2 carbon compound. So can we transfer from these? Yes, this 5 carbon compound. So what will it become? Five minus 2 becomes 3, gap, glyceraldehyde 3-phosphate. But 4 plus 2 becomes So shall we repeat the whole pathway? The whole pathway. You see from your copy, those who have taken enough pain to write, and those who have not taken the pain to write, they do whatever they want. Yellow dress, you have written? Sure? You can see that. Fine. Let us start with 3 molecules of glucose 6-phosphate. The first step. The first 3 steps of this pathway are oxidative phase, rest 4 steps are rearrangement phase. The first and the third step produces NADPH. These 2 reactions are catalyzed by dehydrogenase reaction. The first enzyme is glucose 6-phosphate dehydrogenase, GPDH. The third enzyme is 6-phosphogluconate dehydrogenase. The second enzyme is delta lactonase, which breaks the lactone ring, and in many books it has been clubbed with. The third step produces not only oxidize the substance, but also decarboxylates. So this is an oxidative decarboxylation. So oxidative decarboxylation produces what? The keto sugar ribulose 5-phosphate. Now we begin the rearrangement phase with 3 molecules of ribulose 5-phosphate. We begin the rearrangement phase with 3 molecules of ribulose 5-phosphate. The ribulose 5-phosphate, 3 ribulose 5-phosphates begin with, we have to remember certain mnemonics. The next step involves 3 enzymes, epi, iso, epi. First one epimerase, second one isomerase, third one epimerase. So the first and third ribulose 5-phosphate will convert to xylulose 5-phosphate, the corresponding keto, pentose, and the middle one, the ribose. The middle one will be ribose. So that will be convert from ketose to aldose sugar. So now we have 3 pentose phosphates: xylulose 5-phosphate, ribose 5-phosphate, and xylulose 5-phosphate. In the next 3 steps, there is a similarity. There is rhythm. What is that? Keto, aldo, keto. Transketolase, transaldolase, transketolase. Transketolase, transaldolase, transketolase. Transketolase enzyme always require TPP as coenzyme. Transketolase transfer 2 carbon compound. Transaldolase transfers 3 carbon compound. Correct? So the first reaction, transketolase, occurs between xylulose 5-phosphate and ribose 5-phosphate to produce 5 minus 2, glyceraldehyde 3-phosphate, 5 plus 2, pseudoheptulose 7-phosphate. The next reaction, transaldolase, occurs between glyceraldehyde 3-phosphate and pseudoheptulose 7-phosphate. So transaldolase transfers 3 carbon. So 7 minus 3 becomes 4 erythrose 4-phosphate, and 3 plus 3 becomes 6 fructose 6-phosphate. The fructose 6-phosphate remains as a product, and erythrose 4-phosphate continue for the next reaction, transketolase. The erythrose 4-phosphate is an aldose sugar, so it cannot give away any keto group. So keto group will be taken from the last xylulose 5-phosphate by a enzyme transketolase. So xylulose will give the keto group to erythrose 4-phosphate. So 4 plus 2 will become 6 fructose 6-phosphate. What will be left with? Glyceraldehyde 3-phosphate. Now if we go back, see, this is glycolytic pathway. Can you find out these names? So they merge, they converge with intermediates of glycolytic pathway. So actually it was only 2 steps in glycolysis pathway. Glucose to glucose 6-phosphate, glucose 6-phosphate, fructose 6-phosphate by phosphohexose isomerase. But between these two, one step particular requires so many steps. That's what is called shunt pathway. Clear? Any question? Any doubt? So now you remember. So there are 7 steps in this reaction or in this pathway. This pathway occurs in cytosol. This pathway is very, very important because it pathway is the only pathway to produce NADPH, without which there will be massive hemolysis and patient will die. There are conditions where glucose 6-phosphate dehydrogenase, GSPDH, deficiency is seen. Patient with GSPDH deficiency will succumb to death if any compound or any drug or any substance, any food material they consume which utilizes NADPH more. Well, what will happen? If they are given certain drugs which require NADPH for metabolic reasons, metabolic transformation, then their body will suffer from NADPH deficiency, but their cell cannot produce NADPH much more. So what will happen? They will show hemolysis. They will show hemolysis. That means they will lead to anemia. And even if you treat the anemia, it will come back again and again because it's genetic. So what we have to do? We have to check whether the person, before giving certain drugs, we have to see whether the person is GSPDH deficiency or not. If the person is GSPDH deficiency, we cannot give those drugs. That is a clinical niche, hitch. Right? We cannot give the drug which utilizes NADPH. Fine. So this is a pathway you have to remember. Now why this pathway is important? It is important for several aspects. আমি কি মুছে দেবো? কি significance by importance of HMB pathway. Number one, production of NADPH. Number two, pentose phosphates. That's all. Now, don't have to remember all this. If you know the basic chemistry of biomolecules. We need pentose for what purposes? For synthesis of nucleosides, nucleotides are required for synthesis of what? DNA, RNA. That's all? No, there are plenty. NAD, NADP, FAD, FMN, all these are modified nucleotides. So for synthesis of these also we will require. But we do not synthesize those because we consume, we get it from vitamin outside. For bacteria, they can synthesize their own. Right? So all these are required pentose phosphates. Okay? This is number one. So to synthesize DNA, we need this pathway to run. Otherwise, we will not get ribose 5-phosphate. But anyway, we can get it from the diet. Right? What's wrong in it? We can give vitamin, lot of vitamin. Huh? But the second point is more important. Production of NADPH. NADPH we cannot supplement. We cannot supplement NADPH. Why? Because NADPH is a high energy compound. It will get destroyed as soon as it will get mixed with water. Correct? It is water, it is not soluble in oil also. We cannot give as oil immersion. Difficult because it is polar, not non-polar. Correct? It requires water-free zone, which is possible only inside the protein or lipid membrane, where lipids are not attached to the NADP molecule, but attached to each other, like plasma membrane. Okay? I am telling you very, very critical things, intricate things. So, this NADPH is required for majorly two purposes. Number one, synthesis of a battery of compounds and hormones. For example, cholesterol, fatty acid, then steroid hormones. All steroid hormones require NADPH for synthesis. These synthetic pathways are called reductive synthetic pathway. I repeat, NADPH is required for reductive synthetic pathways. Next, NADPH is required for regeneration of reduced glutathione. NADPH is required for regeneration of reduced glutathione, which is important for maintenance of membrane viability of RBC. RBC membrane come across a lot of oxidative stress because it exchanges oxygen through it. No other membrane exchanges so much of oxygen through them except RBC membrane. So everything gets oxidized. Correct? They suffer most from the oxidative stress. Which one? Erythrocyte. To maintain the membrane integrity of the RBC membrane, we need to remove these reactive oxygen species continuously. And the process utilizes reduced glutathione. So reduced glutathione concentration in the cell decreases. NADPH is required for production of reduced glutathione from oxidized glutathione. What is? This is oxidized glutathione. So from here to correct? Enzyme is glutathione S reductase. Write down this. This particular reaction is so important that without this, RBC will undergo hemolysis. And to run this, we require continuous supply of NADPH, which is possible by only HMP shunt pathway. Hello? Next, cytochrome P450 monooxygenase. This is a system present in the liver or some other tissue which is involved in detoxification of drugs, paracetamol, azithromycin, soframycin, whatever is there in our body, it undergo metabolism and it is detoxified and then excreted through urine. It's a normal process. But there are certain drugs which require hydroxylation for detoxification. These particular drugs utilize NADPH for hydroxylation via cytochrome P450 monooxygenase system. See, I told you there are certain drugs which consumes NADPH more. So the person suffering from G6PD deficiency will succumb to hemolysis. Correct? will succumb to hemolysis. Why? Because he is deficient, he is producing less amount of NADPH, and suddenly you gave a drug which require NADPH for detoxification. So it will utilize all the NADPH in the body, and cell will die due to absence of NADPH. Correct? So it will cause tissue damage, kidney damage, liver damage, not RBC damage, but kidney damage, liver damage. That's why you see, to avoid such this thing, we always go for testing GCPD's activity, particularly in pregnant women. Before giving some drugs, very few drugs can be given pregnant women, like this. So that's all for this chapter. There is nothing to tell further, because I gave you most of the information. It's up to you how you digest it and how you remember, how you reproduce during the examination. Now, for those who think I have missed something, these are the slides. These are the organs which produces these compounds which require NADPH. Tissues with active pentose phosphate pathway. See, adrenal gland for steroid liver for fatty acid and cholesterol biosynthesis, testes for steroid synthesis. Testis synthesizes what? Testosterone, androgens, right? Adipose tissue, fatty acid synthesis, ovary, steroid synthesis, estrogen, progesterone, all these steroids, they require NADPH. Mammary gland, fatty acid synthesis. Why? Because mother's milk contains fat. From where the fat will come? The mammary gland itself will synthesize the fat and introduce in the milk. Red blood cells, nothing, it produces nothing. It maintains. Used up for, in RBC it is used up for maintenance of reduced glutathione or membrane integrity. This is structure of NADPH. These are the reactions which I took pain to draw on the board. I could have gone reading these slides. See, this is a delta lactone. Yes?